Hello, welcome everyone. Hope you had a great lunch and a great poster session. Myself, Susmita Sahu from Icahn School of Medicine, Mount Sinai, and my co-moderator.
John Cook, Houston Methodist. And we're about to begin this session now, so we'd like everyone to take their seat. We got some great talks and already running a little bit behind, so please grab your seat and we'll have our first presentation.
Thank you. And I would like to remind the speakers as well as the crowd here that these are rapid fire presentation, and this is the most exciting session of this meeting. Young presenters are presenting. They get 5 minutes for presentation and 2 minutes for questions. So have your questions ready and be very short and brief with your questions, and we'll have about 10-minute panel discussion at the end of the session. So without further delay, I would like to introduce our first speaker, Seitaro Nomura, Department of Cardiovascular Medicine, University of Tokyo. And the title of his presentation is System-Level Understanding of Cardiovascular Diseases.
Yeah, thank you. And first of all, I'd like to thank Dr. Chang and Dr. Fukuda for giving me a chance to present here today. This is the COI to be disclosed. When we see patients with cardiovascular disease, the interaction between genetic and environmental transcriptional factors may important to understand system-level understanding of cardiovascular disease. In my talk and in my presentation, we have 3 short talks about atrial fibrillation, heart failure, and coronary artery disease. First, we performed genome-wide association study of atrial fibrillation in collaboration with Dr. Kaoru Ito in RIKEN to generate polygenic risk score to distinguish atrial fibrillation patients only by genetic factors. And we also combined the data with epigenomic data to reveal that ER-gamma transcription factor directly binds to the AF SNP regions like this. And we performed functional analysis using iPS-derived cardiomyocytes by addition of ER-gamma inhibitor. If SNP target genes were critically downregulated and showed abnormal myocardial contraction. Therefore, we conclude that perturbation of ER-gamma is a cause of human atrial fibrillation. Next hypothesis is spatial temporal mechanism may regulate cardiac remodeling after myocardial infarction. Therefore, we performed spatial transcriptomic analysis using the heart after myocardial infarction. The dotted circle indicates the region of myocardial infarction. The bottom panel shows the clustering by transcriptomic. And in this study, we focused on the mechanism sensing gene modules, which is critically activated at the border zone of myocardial infarction and in the acute phase after myocardial infarction. The data can be combined with the single-cell RNA sequencing to identify CSRP3 is a center gene of the mechanical sensing gene network. This gene actually encodes MLP protein, which are known to be involved in the mechanical sensing in the cardiomyocytes. Actually, we overexpressed the CSRP3 to show that this gene prevents the pathological remodeling after myocardial infarction. Also, we performed spatial temporal spatial transcriptomic analysis after overexpression and knockdown of CSRP3. Overexpression of these genes not only activates CSRP3, but also activates mechanosensing gene modules. And in contrast, knockdown of this gene directly regulates mechanosensing gene modules. Therefore, CSRP3 is an upstream regulator of this gene mechanism. So we found the spatial temporal mechanosensing mechanism after myocardial infarction. Final talk is about heart failure. We performed pressure overload in the heart and extract single-cell RNA sequencing to generate cell-cell interaction network in the heart, and found that HTR3 is located at the center of cardiac fibroblast network. We deleted HTR3 in cardiac fibroblasts. We extracted cardiomyocytes and investigate the single-cell transcriptomic data. This HTR3 knockout cardiomyocytes show the activation of TGF-beta, DNA damage, and p53 signaling. And in summary, HTR3 in cardiac fibroblasts regulates DNA damage and p53 signaling in cardiomyocytes via TGF-beta signaling. So we skip this slide, and in summary, several factors regulates DNA damage in cardiomyocytes. This is the one potential regulatory pathway in heart failure. Finally, I would like to thank all my collaborators, especially for Professor Issei Komuro, who is my mentor. Thank you.
Thank you, Seitaro, for that excellent presentation. Are there any questions? So I have a question for you. How did you knock out specifically in cardiac fibroblast that particular gene that you mentioned, CHR3? I forgot the name.
A cardiac fibroblast gene?
Yeah, you said that you knocked down, specifically knocked out cardiac fibroblasts, so which construct?
This is not specific. Yeah, specifically expressed in cardiac fibroblasts. So we generated global knockout. Yeah.
Okay.
Yeah.
Thank you. I have more questions, so
Yeah.
I'll follow up. Yeah. Thank you very much.
Thank you so much.
Okay, our next presenter is Jacob Fell from the Stem Cell Unit, Clinic for Cardiology and Pneumology, University Medical Center, Göttingen. He's going to be speaking about LZTR1 deficiency drives heart disease by impairing cellular communication. Dr. Fell.
It's master. Okay. このような機会をいただきありがとうございます。My name is Jakob Fell. I'm a PhD student under Dr. Zügerneck and Professor Zimmermann at the UMG at the Stem Cell Unit in Göttingen, Germany. So our main focus in our group is Noonan syndrome, which is the most common rhizopathy. Rhizopathies are defined as genetic symptoms resulting in dysregulation of the RAS-MAP kinase signaling. Most devastating for the patient is a severe cardiac hypertrophy. And among the genes identified to cause Noonan syndrome, there's also the relatively newly described gene called LZTR1. So LZTR1 has an important regulatory role within the RAS-MAP kinase signaling. It mediates the degradation of its target via ubiquitination, and accordingly, loss of function in this gene leads to an increase in protein levels of its targets, and ultimately the dysregulation of the RAS-MAP kinase signaling. So in a previous publication, we were able to show that patient-derived cardiomyocytes resemble the effect, the cardiomyocyte-specific Noonan syndrome phenotype regarding enhanced RAS-MAP kinase signaling as well as hypertrophic phenotype. So since the heart is a multicellular organ, we wanted to expand our model towards other cell types, namely endothelial cells and cardiac fibroblasts, but in this presentation I will only focus on the fibroblasts. So my project is basically two-sided. On the one hand, I want to see the impact of LZTO1 deficiency on cardiac fibroblasts, and on the other hand, I want to determine the potential impact of LZTO1 deficiency in cardiac cell communication. So what we did is we differentiated wild-type iPS and LZTO1 knockout iPS towards cardiac fibroblasts. And while knockout fibroblasts express regular cardiac fibroblast markers, they are obviously completely deficient for LZTO1. Interestingly, comparing cardiomyocytes and cardiac fibroblasts, you can see that protein levels of LZTO1 in cardiac fibroblasts is increased, suggesting an important role of LZTO1 within the cardiac fibroblast. And as expected, in knockout cells where you have LZTO1 deficiency, the known target in RAS isoform RIT1 is significantly upregulated, or the protein levels are significantly higher. So to further assess the impact of LZTO1 deficiency within fibroblasts, we performed global protein level analysis comparing wild-type fibroblasts versus knockout fibroblasts, and we saw cell cycle and extracellular matrix dysregulation. So therefore, we assessed the proliferative abilities of the knockout cells, and we monitored the increase of confluence over time, and we saw that knockout fibroblasts significantly proliferate faster than the wild-type cells. Strikingly, additional serum does not further increase the proliferative capabilities of the knockout fibroblast, suggesting an intrinsic proliferative force. We also generated three-dimensional tissues consisting of fibroblasts, so-called ECT, so engineered connective tissues. Tissues derived from knockout fibroblasts were significantly more stiff, and this data was further supported by gene expression analysis, where you see that fibrosis-associated genes are upregulated, while more common fibroblast genes are downregulated. So we finally addressed the question of how LZTO1 deficiency impacts cardiac cell communication. We therefore cultured cardiomyocytes in regular culture media as a control in 50% fibroblast media to discount for factors within the fibroblast media itself, as well as wild-type and conditioned LZTO1 knockout conditioned media. So evaluating the effect of the aforementioned media on the cardiomyocytes, we saw that actually LZTO1 deficient conditioned media causes a significant increase in cell size, as in adherence as well as in suspension. And also fibroblast conditioned media triggered a characteristic hypertrophic gene expression pattern. So taking together, we were able to show that LZTO1 deficiency manifests in a cardiac fibroblast-specific manner, resulting in an increased proliferation as well as a pro-fibrotic tendency. More importantly, we could show that LZTO1 deficient fibroblasts trigger cellular cardiomyocyte hypertrophy via paracrine signaling. At the moment, we are actually trying to identify the causing factor within this media. And yeah, I would like to thank you for your attention. Thank you, the organizers, for this amazing event. And thank you, Dr. Lukas Zuganek, for his amazing supervision. And yeah, I'm happy to take questions.
Beautiful presentation. Seeing no one at the mic, let me ask a question of you. If I recall correctly, Noonan syndrome has a vascular condition as well. I think it's a vaso-occlusive, maybe aortic coarctation or pulmonary stenosis. Perhaps this abnormality in fibroblasts also affects vascular fibroblasts? Yes. This is perfectly feasible. I mean, this is perfectly possible. Noonan syndrome has a variety of clinical
implications, basically. Most severe is the cardiac hypertrophy, but we also looked at, for example, endothelial cells. We also saw upregulation of the RAS-MAP kinase signaling in endothelial cells. However, we did not see any significant difference in the wild-type endothelial cell conditioned media versus the knockout.
Thank you. I'm afraid we'll have to move on.
Yes,
Wonderful. We'll
okay.
talk some more.
Thank you.
Do you want to introduce the next speaker?
Our next speaker is Ina Rabinovich-Nikitin from University of Manitoba, Winnipeg, Canada. We'll have the next speaker listed at the end of the session. So Ina, please join here. Oh, sorry. That was my mistake because I thought your slides aren't ready. Sorry for the confusion. I would like to introduce our next speaker, Hidenori Tani, Department of Cardiology, Keio University. And the title of your talk is Production of Mature Engineered Heart Tissues with Heart-Derived Collagen.
Thank you, Chairman. I'm Hidenori Tani from Keio University. Title of my presentation is Production of Mature Engineered Heart Tissues with Heart-Derived Collagen. These are COIs. Immaturity of human iPS-derived cardiomyocytes is a large hurdle. To overcome this, biophysical cues, biochemical cues, and co-culture with no CMs and extracellular matrix are reported to be effective. Though ECM is important, the optimal component of ECM has not been elucidated. So we focused on this ECM and in particular, the most important component, the fibrillar collagens. First, we extracted six organ-derived collagens from adult porcine and generated engineered heart tissue, EHTs, with metabolically purified human iPS cardiomyocytes and each type of collagen. With this EHT system, we could measure contraction force and contraction duration and relaxation duration. We first generated EHTs with each organ-derived collagen without madrigal. As a result, half of the skin collagen-derived EHTs were torn off or deviated from fillers within 2 weeks. But half collagen-derived EHTs kept their contraction and shape. Moreover, calculating the contractile force chronologically, heart collagen-derived EHTs showed higher contraction than other collagens. To elucidate this difference, we compared the profiles of collagen fibrils. As a result, heart, spleen, and kidney collagen produced thinner reconstituted fibrils than liver, lung, and skin collagen. Of note, these trends are related to the composition of the fibrillar collagen rate. In particular, heart collagen, in particular the left ventricle, contained a high percentage of type 3 and 5 collagen than other collagens, other organs, and other sites. So these results indicate that type 3 and 5 collagen may play an important role in retaining tissues. We next compared the EHT with we next compare the maturity of iPS-derived EHT with heart collagen and skin collagen. Under stable EHT generation with madrigal, heart collagen EHT shows higher maturation status in QRT-PCR. Moreover, TEM analysis revealed that heart collagen EHT CM showed more mature organized structures. Furthermore, we analyze the OCR of dissociated cells from each EHT, and hard collagen EHT-derived CMs shows the highest OCR. We next compared the EHT with each purified type hard collagen. We measured relative contractile force and contraction duration and relaxation duration and ratioed them. Commonly, shorter RDCD means an efficient relaxation. As a result, type 1 collagen EHT showed a stronger contractile force, and type 3 collagen EHT showed an efficient relaxation. QRT-PCR revealed that maturation status was upregulated more strongly in crude collagen EHTs than in purified collagens. We finally compared the maturation status of skin-derived purified collagen and that was supplemented with hard type 3 and 5 collagens. As a result, maturation markers are upregulated in mixed type collagen than in purified collagen, and moreover, supplementing hard type 3 and 5 collagen improves the maturation. So maturation may be significantly affected by the type and distribution of collagen. So summing up, my research has led me the following conclusions. Hard collagen plays an important role in retaining J shape and contraction. Hard collagen promoted maturation. Different collagen types have different effects on the contraction relaxation status. And the type and the distribution of collagen plays an important role. Thank you for your attention.
Questions, comments? Wolf, please.
Presentation and very important work. The collagen at the end, you measured the collagen, I believe, when you created the EHTs. And if you do now the analysis after your culture period, is the collagen composition changed or is it the same?
We also check the composition of collagen and from the start and after 4 weeks culture. So collagen was degraded to half after 4 weeks culture, but type 3 collagen retained the amount than type 1 collagen.
Okay.
Thank you.
Dr. Tani, one quick comment. It's not the collagen. I think your data doesn't support that. Let's talk afterwards. I think it's something bound to the collagen that's responsible for the organ-specific differences. So let's talk, but it's not the type of collagen.
Other type of organ collagen? Do you ask?
I think it's something bound to the collagen. I don't think it's the collagen itself, because your data doesn't support that the type 1, the differences in the types of collagen are responsible for the organ-specific differences. But
Yeah,
we'll talk. Let's talk.
Maybe the difference of type 1 collagen organ specificity may exist, but we could not examine the details.
Yeah. Thank you very much for your presentation.
Thank you.
The next speaker is Dr. Ina Rabinovich-Nikitin, Cardiovascular Sciences, St. Boniface Hospital, Albrechtsen Research Center, University of Manitoba, Winnipeg, and Manitoba. She's going to be speaking about retinoic acid-related orphan receptors, regulate autophagy and cell survival in cardiac myocytes during hypoxic stress.
Thank you. I would like first to thank the organizers for giving me the opportunity to speak here today. And the focus of my talk is retinoic acid-related orphan receptors, or RORs, and how they regulate autophagy and cell survival in cardiac myocytes during hypoxia. So RORs are part of the circadian system, and the circadian system is 24-hour cycles that regulate the physiological processes of all living beings. In the heart, circadian rhythms regulate many processes such as heart rate and blood pressure. But circadian rhythms can also regulate pathophysiological processes of the heart. And it is known that different cardiovascular diseases or cardiac events occur in a time-dependent manner with higher incidence in the early morning hours. And we can see the example here, which represents 2 cycles of 24 hours. that shows the incidence of myocardial infarction being increased in the early morning hours between 6 to 10 a.m. and declines later in the day. Now, one of the underlying mechanisms for myocardial infarction is autophagy. And this paper that was published by our lab a couple of years ago showed that circadian rhythm, and especially the clock gene, which is one of the main circadian genes, can regulate autophagy by binding to EBOX elements on different autophagy gene promoters. And when we performed in silico analysis on the different autophagy genes, we saw that they not only have EBOX elements on them, but also ROR elements for binding of ROR, which is represented by the GGTCA sequence. And therefore, in our study, we were interested to look into how ROR can regulate autophagy and cell survival in cardiac myocytes. So for that purpose, we did non-biased screening of a small molecule library, and we identified the molecule nobletin. And nobletin is known to upregulate the expression levels of ROR, which induces activation of the clock BML1 and activation of circadian. So in order to test the role of ROR in cardiac myocytes during hypoxia, we first looked at the levels of ROR during hypoxia. And we saw that ROR is downregulated during hypoxia. But when we treated the cells with nobletin, we were able to upregulate ROR activation or ROR expression in cardiac myocytes during hypoxia. Now, during hypoxia, we also know that there is increased cell death, which is evident by the red cells compared to the green cells. And when we treated the cells with nobletin, we were able to to rescue the cardiac cell death during hypoxia. And in order to show that the cardiac cell survival is also ROR dependent, we inhibited ROR in cardiac myocytes. We knocked down ROR. And we saw that in the presence of SROR, nobletin failed to rescue cardiac cell death during hypoxia. And interestingly, we noticed that even during normoxia, knocking down ROR is causing increased cell death, which means that ROR probably has a role in cardiac cell survival. Next, we wanted to show that ROR is also linked to activation of autophagy in cardiac myocytes. So we checked different autophagic markers such as Becklin, P62, and LC3. And we saw that they were all downregulated during hypoxia. But when we treated the cells with nobletin, we were able to rescue autophagic markers in cardiac myocytes. And when we treated the cells with nobletin in the presence of SROR, nobletin failed to rescue autophagic markers in cardiac myocytes. And this also coincided with the autophagic function, as evident by the GFP LC3 staining. So during hypoxia, we have downregulation of autophagy, but when we treated the cells with nobletin, we saw increased autophagosome formation, which means we had activation of autophagy. And finally, we also wanted to show that the cardiac cell death rescue by nobletin is autophagy dependent. So we inhibited autophagy by SH-ATG7, and we saw that in the presence of SH-ATG7, nobletin failed to rescue cardiac cell death, which also coincided with failure to rescue expression levels of autophagy genes such as LC3, which means that nobletin requires both autophagy and circadian mechanism in order to rescue cardiac cell death. So in conclusion, we showed here for the first time that there is a link between ROR gene, autophagy, and cardiac cell survival. And loss of ROR, or knocking down ROR gene activity, results in increased cell death from impaired activation of autophagy. But restoring ROR activity, such as by nobletin, is able to upregulate autophagy gene expression and increase cardiac cell viability. And therefore, we suggest that different interdictions that can influence ROR activity, such as nobletin, may prove beneficial in restoring circadian function following hypoxic stress. And finally, I would like to thank my lab members, Molly, Ken, and Daria, and the funding agencies, and our collaborators, Dr. Lori Kirschenbaum, Dr. Junichi Sadoshima, and Dr. Gad Asher. Thank you.
Very nice presentation. Open to questions, comments? Do you have, tell us what plans you have to go forward with this in a translational way. Do you have any plans to translate this into a clinical application?
Yeah, so actually circadian defects like we saw here with downregulation of clock or, with ROR, or like we saw before with clock, is evident in models such as shift workers, for example, or jet lag. So if we're able to rescue circadian defects, for example, with nobletin by treating nobletin to mice or even to humans by supplement, that's something that we can use in clinic.
Wonderful. Thank you for that presentation.
Thank you.
Our next speaker is Lee Lai from Department of Cardiovascular Sciences, Houston Methodist, Texas, USA. And the title of her talk is Metabolic and Epigenic Coupled Regulation of Transdifferentiation and Vascular Regulation.
Good afternoon, everyone. Today is my great honor to talk about some of our lab's work in this wonderful meeting, which is about a novel vascular regeneration mechanism. So the mechanism I'm talking about is called angiogenic transdifferentiation from fibroblasts to endothelial cells. And in our theory, the fibroblast cells could significantly contribute to the repopulation of endothelial cells under the stimulation of angiogenesis. So here are some evidence showing the natural occurring process in the level of ex vivo and in vivo. So iodic ring assay is a classic angiogenesis function assay in which we will cut out the aorta from the mice and then we cut the aorta tissue into ring structures and then we embed them in the collagen. And a few days after the embedding, we can clearly observe the sprouting of the microvasculatures, which are mainly comprised with endothelial cells. So to study whether the fibroblast cells could contribute to this angiogenesis process, we utilized the aortic tissue from the lineage tracing, fibroblast lineage tracing mice, and we performed this assay. And from the immunofluorescence study, we can see that we can track down the fibroblast lineage cells with YP positive signals, and we can also observe the CD31 positive endothelial cells. So what is the most interesting for us is that we can clearly identify some double positive cells, which we believe are the induced endothelial cells that are transdifferentiated from fibroblast cells. And those double positive cells are actually incorporated into these microvessels, further suggests that this angiogenic transdifferentiation could contribute to functional angiogenesis. So we further utilize this similar lineage tracing strategy and perform some in vivo vascular regeneration models, which is a high limb ischemia model. So in this model, we ligate the femoral artery in the limb, and 21 days or so, we can observe a fully reperfused limb vasculature. That's why we call it the vascular regeneration, because it really recovers. And by this model, we can clearly detect that there are CD144 positive endothelial cells that also has YAP signaling, suggesting this angiogenic transdifferentiation could actually contribute to the vascular regeneration. So to better understand and manipulate this endogenous healing process, and potentially could use this mechanism to treat the vascular disease or enhance vascular regeneration, I decided to look into the influence of metabolism during this process. As it's well known that many metabolites or metabolic enzymes they are known to modify many key epigenetic factors and then regulate cellular plasticity. So indeed, my initial findings show that during the transdifferentiation, there is acute metabolic switch away from oxidative phosphorylation towards glycolysis, which is associated with accumulation of a citrate, which is a TCA cycle intermediates. And those upregulated citrate can be shunted out of the mitochondria and towards the nucleus, where it can be further converted to acetyl-CoA under the control of ATP citrate lyase, ACL or ACLY, and those enhanced acetyl-CoA production are, enhance the histone isolation, DNA accessibility, and also the transcription, which are all required for cell phase transition and transdifferentiation in our case. So since we know the metabolism could play an important role, so to formally characterize the metabolic components during this novel healing process, I used a on-targeted metabolomics to determine additional metabolic determinants of transdifferentiation. And with this assay, with this screening, we found the most upregulated metabolites during this modification is called UDP-GLICNAC. So what is UDP-GLICNAC? So UDP-GLICNAC is an end product of a hexosome biosynthesis pathway, and it's also the substrate for important post-translation modification called O-glycosylation. And a pair of enzymes are responsible for this modification. OGT is the enzyme that is responsible for adding this modification, while the OGA is the enzyme that is responsible for removes this modification. So to further determine whether this organization modification could have a potential role in the trend differentiation in vivo, we utilize a similar linear tracing mice, which is FSP1 fibrosis-specific protein Cre mice, and we performed the high-limit ischemia model with those mice with or without the treatment of OSMI, which is the organization inhibitor. And first of all, based on the Doppler imaging result, we showed that the OSMI treatment could impair the overall vascular recovery from the ischemia. And if we isolate the cells, the non-myocyte cells from the ischemic limb tissue 7 days post-surgery and characterize those cells with flow cytometry, we can see that among all the CD31 positive cells, in the sham mice, we don't see a population that has YIP positive signal, suggesting there is no baseline reaction before surgery. And what is more important, in the vehicle-treated mice, we can observe a well-separated major population that is YIP positive. And this population is what we consider as a trend differentiation population. And also based on the size of this population, we believe that this trend differentiation process is significantly contributing to the repopulation of the CD31 positive endothelial cells. And what is the most important thing for us is the OSMI treatment could actually largely diminish this population, further suggest that this organization could promote vascular recovery through trend differentiation mechanism. To further prove our hypothesis, we generate the conditioned knockout mice to manipulate organization in the fibrous cell lineage cells and perform high-limit ischemia with those mice. So we detect that in the OGT fibroblast-specific knockout mice has a reduced vascular recovery, while the OGA fibroblast-specific knockout mice have an enhanced vascular recovery, further support our hypothesis here. So
could you kindly summarize, please?
Yes, this is summarized. Yes. So with that, I hope you can get my take-home message that the androgenic trend differentiation could be a significant mechanism for the vascular regeneration, and the modulation of the metabolic aeration could regulate androgenic trend differentiation and ultimately vascular regeneration. So with that, I would acknowledge my mentor, Dr. John Cook, and also the CVBE meeting organizer, Dr. Jay John and Fukuda, and all the meeting committees to give me this precious early career investigator award. And I will be happy to take any questions.
Sorry, in the interest of time, we'll have to move on. Sure. Thank you for the excellent work.
Our last speaker of this session is Kay Wong from North Carolina State University, who will be speaking to us about a microneedle patch loaded with IL-4 macroparticles facilitates heart repair through cardiomyocyte phenotype transition. Dr. Wong.
Thank you, Dr. Cook, for the introduction. Yeah, so today, when you see this topic, you probably will ask several questions. First, why IL-4? Second, why using this microneedle patch? So let me explain the rationale. So IL-4 has been largely used in vitro tests to polarize macrophages. Because macrophages, as you know, every kind of injury macrophage probably is a first soldier in the front line. So the macrophage change is also the key to tell whether this tissue will go worse or it can go to the healing process. So we want to see, okay, whether this IL-4 can be delivered in vivo to see the same result, whether this early immune modulation can causing some difference or the treatment effect for the myocardial infarction. So in this process, it has some tricky things. So IL-4 has a very short half-life. It's about 15 minutes. And also, systematic delivery of IL-4 can causing other problems. That's why we're trying to using this delivery method to locally deliver IL-4 to the injury site. So previously, we have generated this macroneedle patch to deliver the proteins, and so that we want to use the same method, but difference is this macroneedle patch is dissolvable, and we loaded this IL-4 macroparticles inside of the patch. After we put it on, this patch can be starting dissolve into gelish format in 5 to 10 minutes. And this part is fully supported by Dr. Song Li from UCLA. Then we tested in vivo in the rat heart because rat heart is relatively larger and it's good for patch delivery. And we see some functional benefits and morphological benefits. And from this interesting staining, we find that actually the histone 3, PH3, has expressed in the cardiomyocytes in vivo. And also, however, we didn't find any Ki67 expression in cardiomyocytes yet. So we want to see why this early immune process can causing like a proliferation of the cardiomyocytes. We did the single nucleus RNA sequencing at day 3 after application, and we find that the major thing is the cardiomyocytes. And these cardiomyocytes are mainly coming from the cardiovascular. So we find that the major one's difference is from the cardiomyocytes type 4 to type 2. Type 2 is mainly inside of the IL-4 treated group, and type 4 is mainly in the MI-only group. And from the trajectory map from A and B, we can see that is a little bit swift from the X to Z axis. And the highly differential expressed genes can show that these genes are actually showing the new cardiomyocytes phenotype 2 has a development process and growth. So we're thinking, okay, this is whether this related to macrophages. So we get all the macrophage done, and the macrophage shows not really M1 or M2, but it has a gene expression of the more anti-inflammatory after treatment, and the geo enrichment showing that is negatively regulates the immune response. So we're thinking in this anti-inflammatory environment whether these cells can communicate differently. So we did the cell-cell communication, and we found that actually you can see that epithelial cells they're more or quiescent, and also from the cardiomyocytes cross-talking, we saw that the new phenotype after treatment is like the old cardiomyocytes. They respond well with inflammatory response signaling, and also with the epithelial cell, endothelial cells, they respond with NPR1, which is maintain the endothelial cell into a homeostasis state. And also with CM4, which is mainly in the MI group, they are more fibroblast-like. They reject to receive any any signals, but sending out a lot of like collagen and the laminin and also the APP signaling. So at the same time, we're trying to see if this result could be seen in larger animals. So we put a larger patch on the pig heart, and it shows very nicely. We find some Ki67 expression in the cardiomyocytes. This is a summary page. I will just skip that. For future directions, does the IL-4 change the CM directly or indirectly? Because we find that the cardiomyocytes can show some IL-4 receptors. Another thing is whether this signaling pathway is related to cardiomyocytes is which pathway is most related. Right now we have some hints, which is PI3K, AKT pathway, FOXO pathway, and MAPK pathway. With Dr. Cook's suggestion in the poster session, we will definitely do some cardiomyocytes plasticity test and also further DNA level changes. And also from the larger animals, we will do more treatment efficiency, safety, and feasibility test. And that is all. This is our group. Thanks the organizers, Dr. J. Zhang, Dr. Fukuda, to invite me to this meeting. Learned a lot in this meeting, and also thank my mentor, Dr. Ke Cheng, who has been helped me since I was a grad student, and our lab members closely worked with me, and definitely our collaborator, Dr. Song Li from UCLA, and Dr. Kel Marcios, who is the main surgeon for the pig study. Thank you.
Well, in the interest of time, we'll move forward with the panel discussion. We're supposed to have a panel discussion now, and that includes Dr. Wolf Zimmerman and Dr. Zhuang and Chong. And I think if you want to step up to the microphone, maybe just give us, we don't have much time before the break, maybe just give us a reaction to what you heard. What was the single most important thing you heard during these presentations, or any questions for the speakers? Maybe I'll start with Dr. Hidenori Tani, his presentation. You can go to a microphone so you can respond if you like, Dr. Tani. I made a cryptic remark about your presentation, which I thought was very interesting. You showed us that there was organ-specific differences in the response of cardiomyocytes. And the heart matrix, the collagen derived from the heart matrix, seemed to be most supportive of the cardiomyocytes. Skin was least. Lung actually seemed to be quite good. My comment related to your analysis of what the differences were between cardiac and skin and lung. You looked at the different types of collagen, type 1 and 3 and 5, and but the differences there were very modest, and in case of skin, it was no different than lung, but they were very different in terms of their support of cardiomyocytes. I suggested that maybe there was something else that you're missing there, and I suggested to my colleague here that it might be exosomes that are bound to the collagen. Very hard to get rid of them. We took some processed collagen from commercially available collagen sheets that are used for wound repair. And we did microRNA profiling of these. These are heavily treated, processed with acid and heating. And there was still microRNA there that was biologically active. So I wonder, can you react to that? Any thoughts about how to understand these differences further?
Thank you. we microarray the difference of collagen with EHT collagen. EHT with skin collagen and heart collagen only two groups. So we still have not the answer. So we want to examine in the near future.
If I may comment, and it's excellent work, and that's why you are receiving more questions. And I have a question a little bit different. So in myocardial infarction, we have all these fibrotic tissues which secrete a lot of extracellular matrix material. So when you are trying, the implication, if I understand correctly from your study, is like collagen. Application of these external factors would help the cell adhere and maybe mature, or like in that line. Is that correct? So there is pre-existing collagen and extensive secretion during this remodeling phase. So how do you anticipate your cells behave in that microenvironment? Do you have any thoughts?
So microarray result between skin and heart collagen? I'm sorry, I could not get it.
Okay, we'll chat later on.
Yeah,
Thank you very much. And we have a few more minutes left, like 5-7 minutes. I would encourage if anyone else have any question for the presenters, please come up to the microphone and initiate a conversation. So I would like to start with a question to Jacob Well. Yes, please, if you can come to the microphone. So your interesting observation in the knockout, fibroblast knockout in LZTR1, that's the model you are using, and you found out that increases proliferation in fibroblast, but increases hypertrophy in cardiomyocyte. So why not proliferation in cardiomyocyte? Any thoughts on that, and if that can be used for some therapeutic targeting and things like that?
Yeah, well, it's a very good comment. We actually assume that LZTO1 defines its dysfunction because of the RAS degradation. And we actually think that the cell-specific manifestation of the phenotype is largely dependent on which RAS isoforms are actually present and bind to LZTO1. So for example, in cardiomyocytes, we assume that the primary target is MRAS rather than RIT1, although RIT1 is also involved. So yeah, we assume that this is a cell-specific thing. But if the second point for the translational point is, I mentioned that we are in the process of identifying the contributing factor or causing factor of this cardiomyocyte hypertrophy. We are not so sure yet, so we don't want to disclose this as of now. But the most straightforward approach would be obviously to find inhibitors, specific inhibitors for this specific cytokine, and hopefully we can actually decrease the disease load for the patients after all.
Thank you. Thank you very much.
Well, I would like to just take the opportunity to make a comment. So this session is designed for the early career young investigators to practice your presentation with given time window. So 5 minutes, that's it, to convey the message. The most common problem for a young investigator, myself included when I was young, is try to tell too much. And so 5 minutes, 10 minutes, 20 minutes, it's a skillful thing for you in your career. You will experience lots of occasions. We will test your skill to convey your message with time window. So science is great, very encouraged, but there's some work to do. And this is not only for the young investigators. We see 20 minutes presentation, many of them fail to cut within 20 minutes. So we all have work to do, myself probably included also.
Thank you, Dr. Zhang, for that crystallized wisdom. And I think we'll now end and go to our coffee break. Thank you for all the presenters and the questions.